So after preparation of media plates with the help of Muller-Hinton agar media, we are going to find out that whether we have enough amount of our bacterial culture or not. So that will be done with the help of young culture of Bacillus megaterium and Escherichia coli are taken, and then after they are standardized for their turbidity. The standardization of turbidity will be done with the help of 0.5 McFarland standard. The standard is prepared from barium chloride, and with the help of this standard we can standardize our turbidity of culture in such a way that we can get enough amount of growth in the form of loan in the entire plate. So that is our purpose. Now after adjusting the turbidity of our young cultures, then after we proceed for preparation of seeded plate. Now how it is to be done? First of all we take Muller-Hinton agar plate, Muller-Hinton agar media. Okay, it is already prepared, now solidified, and in that we will put a drop of bacterial culture with the help of micropipette in the center of the plate, and then after it will be spreaded with the help of a spreader which is sterile one. Okay, now the entire drop will be spreaded in the uniformly in the plate like this. So it will cover entire plate like this, and then after this plate is called seeded plate. Now in this seeded media, now we are going to add the crude fermentation product. How? So in seeded media, with the help of cup bearer, we will make the wells. So 4 wells we are going to make in this. So here 1, here 2, here 3, and here 4. So we are going to make 4 wells in which we are removing this agar material completely from this particular region. So now it is empty and it can be filled with something. So now with the help of micropipette in these wells, we are adding 20 to 50 microliter of our crude fermentation product. Now after adding in four of these wells proper amount of this crude fermentation product, this plate is kept at 37 degrees centigrade for 24 hours for incubation. Now after incubation, we are going to check for the incubation activity. How? We are going to measure the zone of incubation. Now that will be with the help of a scale in millimeters. Now here also we are doing one other thing, that is we are putting positive control and negative control. Positive control and negative control. Now what is it? Now in positive control we take the standard antibiotic with our test sample, and we also here get zone of inhibition which is standard. And in case of negative control, we only add bacteria, but we are not adding anything like antimicrobial compound, not standard antibiotic and not crude fermentation product. So what will happen in negative control plate, we will have complete lawn formation like this. So we can be very sure that culture is okay, it is young and it has given enough of the growth. Okay, so it is confirmed now. But with the standard plate, that is with the positive control, what we are going to do, we are going to compare our results, which will be in the form of incubation zones. Okay, here. And we are going to compare our positive control results with these particular results of crude fermentation product, and we will try to understand, try to evaluate that whether our crude fermentation product is having any microbial potential or not. Okay, is it having more amount of inhibition than standard antibiotic, or it is having lesser amount of inhibition than our standard antibiotic. And inhibition, you can say level of inhibition will be found with the help of the size of the zone. If the size of zone of inhibition is very big, it means that they have inhibited bacteria in the enough amount. So they could not grow properly, and it has given very clear bigger zone. So if the zones are smaller, it is considered that inhibition activity is less. Okay. So in this way, we are going to find out the inhibition and antimicrobial activity in crude fermentation. Now, before we take the crude fermentation sample, we have already centrifuged it, and then after we have filtered it. So during centrifugation, what we have done? During centrifugation, we have filled the cells, and then after we have filtered it out. So what we have done? We have taken out the cell debris. So what is there in the filtrate? Only antimicrobial portion or antimicrobial compound. Okay, so and this filtrate we have taken in our practical for our crude fermentation sample. Okay, now why we do this particular experiment? The reason is we can find out a new drug, we can optimize, or we can have optimization of production of these particular crude fermentation sample or crude fermentation product. And the third thing is we are able to treat as well as we are able to prevent the diseases caused by microorganisms with the help of these crude fermentation product. Okay, so this is the feature of this practical, and it is really very important for downstream processing.